Thank you. It's great to be here. Fantastic talks, great start of the day. So I'm Livnat Jerby. I'm an assistant professor here at the Department of Genetics. And today I'm going to tell you about some of the work that we've been doing on mapping and attempting to redirect cell responses and location. And so in some ways, this is going to be a little different in the sense that spatial biology is very complex and very multifactorial. Of course, we have better tools to describe what we're seeing, but ideally, we'd also want to have a better understanding of cause and effect of mechanisms. And I think this is where we're still a little bit behind. Sorry. Okay, here we are. So when we think of any process, physiological process, whether it's TB, prostate cancer, immune responses, or many other processes, pick your favorite, they often occur across different levels of regulation, different scales. Whether we're thinking of the cell as an autonomous computing unit, interactions between cells, or emerging properties that occur both at the tissue and the whole body level. And ideally, we'd want to understand how everything is interconnected, and we have more powerful tools to be able to do that. I'm going to talk a little bit about the work that we've been doing to get to where we want to be in terms of technology development. But on the flip side of it, also, we know that many times processes as immune responses are becoming dysfunctional. And as we're experimenting and have the ability to experiment at a greater scale, one of the things that we really enjoy doing in my lab is to also put on our engineering glasses and ask whether we can probe the system, whether we can augment, redirect, or maybe even generate new types of immune responses that are going to be more targeted and more amenable to our control, potentially provide a basis for new types of disease treatment and prevention. So with that in mind, today's talk will have two to three main parts. And I'm going to give more of an overview and a bird's eye view of the type of projects that we've been doing in this space. And as you'll see, each one of them has a spatial component to it. So we're going to start not with what we see in the lab, but what we see in patients, and specifically ask, what can spatial organization tell us about immune evasion and cancer? We're then going to combine this with high content CRISPR screens that we can do in highly controlled model systems. And I think that it's really together, these two, that can allow us to identify new types of immunomodulating mechanisms. Then in the second part of the talk, I'm mostly going to focus on our work in the context of cell design and our attempt to modify immune cells to sense cancer cells in the new and more precise ways, and in ways that will allow us to mobilize these cells, these engineered immune cells, or potentially also endogenous immune cells, to specific locations in the body. Because location is really key, and I think that we can control function if we can control location when it comes to immune cells. So when I started my lab here, one of the things that we set out to do, and is still a major objective of the lab, is to have a better understanding of what is preventing anti-tumor immune responses in patients. Because there are many things that we can model in the lab and we can set out to target, but they might not recapitulate what is really the barrier in patients. So when I was a postdoc in the Vivlegov's lab back at the Broad, we did some of the early work using single-cell genomics for that purpose. As some of you might know, actually the approval of immune checkpoint blockade was shortly before single-cell transcriptomics and single-cell genomics took off. So we were able to start addressing this and having some better resolution to study what is happening in patients. But it was also clear that if we don't know where immune cells are, if we don't know where different cells are, it's going to be very difficult to understand how they regulate each other. And so in a study that was led by three members of my lab, Carmen, Olivia, and Christine, we set out to use different spatial transcriptomics technologies that now allow us to profile hundreds to thousands of different genes at the single molecule resolution. And in this case, we focused on ovarian cancer, profiled millions of cells, 130 tumors, and across more than almost 100 patients. And with these large patient populations, we can now also start correlating what we're seeing with genetic variation and with clinical outcomes. So because this study is already published, I'm only going to give the main takeaways. I think one of the main things that we learned is that tumor genetics, and specifically the copy number alterations of the tumor, are tightly linked to malignant cell states. This is something that we've known for a long time, but here we also observed that these same malignant cell states were marking and predictive of whether T cells and NK cells are going to get recruited and infiltrate into the tumor. And as we then went on to show, they're also predictive of whether a patient is going to respond to immune checkpoint blockade. Now, if this is a causal link, we cannot change a patient's genetics, right? But what we can try and do is have interventions that will have a similar effect. And if we find these interventions and we show that they make a difference, then perhaps this further supports the causal link and the notion that there are certain genetic drivers of immune evasion versus recruitment. And so this is exactly what we did. We identified gene knockouts and inhibitions that change malignant cell states from what we proposed as a non-immunogenic, non-responsive to responsive cell state, and showed that this now allows both T cells and NK cells to better recognize these cells and eradicate them. Now, this wouldn't have been possible just based on the spatial data. Even though it was unique and it was quite comprehensive, it still wasn't enough, because when we look at transcriptome, we're not observing latent regulators that might be pulling the strings. And so this is where Perturb-seq comes in. Perturb-seq is a method that we developed back when I was at the Broad. It was developed in Aviv Regev's lab, where we essentially turn each cell into its own little experiment, and we can perform pooled CRISPR or pooled genetic screens where we profile each cell and combine this with single cell RNA sequencing readouts. And so in this case, we use Perturb-seq to identify latent cell state regulators, and also used previous Perturb-seq screens in order to perform data-driven experimental design and to narrow down the search space up front. And this was really critical. Now, another approach that we've been taking is quite orthogonal and synergistic to what I described so far, where we're using highly controlled model systems, as the one shown here, both ex vivo and in vivo, where we can clearly distinguish between on and off target effects of the anti-tumor immune response. And so what you're seeing here in red are CD8 T cells, primary CD8 T cells, that we transduce to express a specific T cell receptor, or TCR, that allows them to sense those cancer GFP positive spheroids. And as you can see, they're kind of get recruited and latch onto them, resulting later on in, I don't know if I have, oh, okay. So resulting later on in cytotoxicity. And as you can see, if the CD8 T cells do not have that TCR, we don't see that spatial patterning of recruitment. But the important thing here is that now we can use these types of models in combination with perturbations, whether it's CRISPR knockout, activation, ORFs, et cetera. We can probe different cells and we can ask not necessarily how are we going to make the immune response more or less active, but how can we make it more precise and more on target, which I think is really the key, no matter which disease or pathology we're talking about. So, for example, in a study that was led by Risa Khanna, a student in the lab, he performed CRISPR activation screens in melanoma cells that he then exposed to either targeting CD8 T cells or non-targeting CD8 T cells. And with these types of screens, we can now detect sensitizers or desensitizers for TCR-dependent cytotoxicity. And when we do this, we identify dozens of different regulators, as shown here. And these mark immune evasion mechanisms that depend not on the function of proteins, but on deficits in RNA levels of different genes. And so one of the key takeaways here is that we cannot target those types of immune evasion mechanisms with standard conventional drugs, which are either agonists or antagonists, and therefore work at the protein level. And for that reason, we're now working on a variety of ways to deliver RNA and to translate these findings to RNA-based interventions. What we would also love to do is, given that there are so many regulators and that we have the ability to discover them with these types of screens, it's a little dissatisfying to just follow up on one or two top hits. And so we would love to follow up on all hits and do this mechanistically. And for that purpose, we developed an in situ perturb-seq method where we can detect with very high accuracy different genetic perturbations in situ, whether guide RNAs or ORFs, and do this in conjunction with single molecule resolution detection of, again, up to 6,000 genes, in either spatial transcriptomics or spatial proteomics, because this is compatible with commercially available platforms of spatial transcriptomics. And so now with these types of tools that we applied also in vivo, as shown here, we can perform pooled screens where we have many different types of perturbations, dozens to hundreds of perturbations, and we can observe not only how they impact the cell that we perturbed itself, but also the cells around it. And particularly in this case, we would want to know how perturbation of a cancer cell then goes on to impact T cell recruitment, T cell suppression, exhaustion, etc. So these are results that we actually just got last week, but you can see an entire tumor here where we're marking cancer cells in black, and specifically cancer cells with a specific perturbation, for example, a CD44 ORF in red. You can see the expression of CD44, and here you can see in a similar way the detection of a MYC ORF and the overall expression of a MYC transcriptional program that is matching it. And one of the important things to note here is that unlike most pooled CRISPR screens, where we are sometimes concerned about cross-contamination across different perturbations, we do see that spatially there is a segregation. There are different zones where there are different perturbations that are most dominant. And this again will allow us to properly map how the perturbation is impacting the tumor microenvironment or the cells around the perturbed cells. And we're now also taking this to the next level by perturbing different cell types in the same experiment and combining this with a variety of machine learning algorithms and tools, including Dialog that we developed at the time to identify multicellular programs, but this time also having the perturbations to actually be able to look at this more mechanistically in order to parse these types of circuits. And that brings me to our work in the space of immune cell engineering, where we're leveraging the fact that we can take immune cells out of blood or other tissues or biofluids. We can then modify them in a variety of ways by turning different genes on and off or adding synthetic proteins, and then study their function either ex vivo or in vivo. So with that type of scheme that can also in principle be applied clinically, we can ask very basic questions. We can ask how can we optimize different functions of interest. We can ask whether we can give immune cells new features that they don't have naturally. For example, can they carry drugs? Can we combine two different types of cells so that we can kind of get the best of both worlds? And can we start modifying cells not as separate entities, but as different units within larger multicellular machinery that can potentially carry out more complex functions. So for the rest of the talk, I want to focus on a particular feature that we're very interested in, which is the ability of immune cells to move around and to get to the right place. And I would argue that there are many diseases and pathologies that are going to be mitigated if we will be able to control immune cell location. Now, I also often say that immune cells versus other cells are kind of like animals versus plants. The fact that they can move around means that they need to make decisions and calculations that I think are really fascinating. So some time ago, I think we actually don't have, oh, okay. I have a slide. So some time ago, we started thinking more deeply, what is it that controls cell location? And to what extent can we redirect cells to specific sites? But before I'll tell you how we're probing this, I do want to go back to our spatial study that I presented in the beginning and show you what we find there. But this time, when looking at this from the perspective of CD8 T cells. So here you're seeing a UMAP that is based on the gene expression of CD8 T cells. And as expected, we find different well-established CD8 T cell states. But now we can also ask, with the spatial data, we can ask, where are these T cells? And when we do this, we find that only specific clusters based on transcriptionally defined clusters are those that infiltrate into the tissue. So here, each dot corresponds to a CD8 T cell, and the color denotes to what extent did it infiltrate into the malignant areas of the tissue versus the outskirts, which are more where we find stroma. So based on this information, we were able to identify and characterize the T cell signature that allows us to predict whether T cells are infiltrating or not. And you can see this is unseen test data where it's shown in situ, where the CD8 T cells are colored based on the expression of this transcriptional program. And as you can see, those that overexpress it in red are those that infiltrate into the malignant areas shown here in gray. And those that underexpress it are those that are at the outskirts and are next to stroma shown here in white. So some of the genes in the signature are beginning to indicate that T cells up front are different and make different decisions in terms of whether or not to get into the tumor. For example, we see the upregulation of CXCR6 matching the CXCL16, which is expressed by the malignant cells, as well as the downregulation of CXCR4 matching the CXCL12 that is expressed by fibroblasts. But now we can ask, to what extent can we modify this? Are we talking about mostly chemokine and chemokine receptors? There's a lot already known about immune cell migration. So is it all there is to it? And maybe if we already have all the mapping, then we can start using it in new and more powerful ways. So we have a variety of studies in the lab that aim to address this question. I want to tell you about one of them that is led by Youngmin Kim, a postdoc in the lab, where he introduced the CRISPR-Cas9 SAM system for CRISPR activation in natural killer cells or NK cells. He showed that he can use this form of gene activation to robustly activate different genes. And if this gene is a receptor, that receptor will also get to the membrane as shown here. And also if we activate a chemokine receptor, for example, the IL-8 receptor, then we redirect the NK cells towards that chemokine, the IL-8. Okay, so this is all very nice. The system is working, but now what can we do with this? Now we can probe NK cells in hundreds to thousands of different ways, and we can track where they're going, sort of let them compete against each other to optimize their ability to migrate to specific sites. But before attempting to test this in more complex systems, we started with something that's way simpler, which are these cancer spheroids such that we can gate on the perturbed NK cells that migrate into the spheroid and sort of select for them in a screening setting. And when we do this, we identify several very strong hits that repeatedly appear. And interestingly, most of these genes are not known for their role in NK cell biology, and they're not known for their role in NK cell migration. And this is perhaps not surprising because many of these are not even expressed by NK cells. For example, when we look at single cell data from breast cancer patients, we find that these genes are overexpressed in immune cells from tumor samples compared to those in blood samples, but mostly in myeloid cells and dendritic cells and even neutrophils. So this is suggesting that perhaps we're rewiring the system in a way that we're taking a cell type invariant property of a gene, and we're kind of plugging it where it shouldn't be, but maybe could be beneficial under certain circumstances. Okay, so what are these genes? It turns out that they're all G protein coupled receptors, or GPCRs, which means that they have a very similar structure to that of chemokine receptors. But unlike chemokines, these receptors, most of them are known, other than C5AR1, are known to respond to different metabolites, primarily lipid and cholesterol derivatives. And so we then went on to show that once we activate those receptors in the NK cells, we can now redirect them not to chemokines, but to these specific metabolites. For example, the cholesterol, oxidized cholesterol derivative in the case of GPR183. So lastly, we set out to study this in vivo. Obviously, when we're performing these screens and spheroids, that's very nice, but obviously it doesn't capture the full complexity that NK cells have to deal with when they're migrating into the tumor. And so we kind of switched gears to performing these types of screens in mouse models, either to gate on migration towards different organs or different sites, or to different tumors with different molecular and mechanical properties, both breast cancer and ovarian cancer. These are studies that are ongoing. But I want to show you the results that we have from the breast cancer CRISPR activation NK screens, where again, we're identifying NK cells that are better at migrating to these breast cancer tumors. And when we do this, our very top hits are exactly the same ones that we identified ex vivo. I don't know if you have good visual memory, but these are the very same genes, with GPR183 also coming up as a top hit in our ovarian cancer screens. And so lastly, and these are still ongoing experiments, we have initial data suggesting and showing that once we activate GPR183 in NK cells, these NK cells better control breast cancer tumors. These are non-specific NK cells, and so we're now testing this with CAR NK cells and with CAR T cells as well. Okay, so to summarize, I want to both give some summary and conclusions, but also a little bit of forward looking in terms of where we want to be and where I think this is heading. So first, I think the study that we just published of ovarian cancer is setting the stage for mapping and potentially also targeting the genetic basis of immune evasion. There have been massive studies to understand the genetic basis of tumorigenesis, to identify tumor suppressors and oncogenes. And I would argue that similarly, there are different immune regulators, positive and negative, that become dysregulated in cancer for a variety of reasons, including genetic mutations and copy number alterations. And if we'll be able to make those connections, we'll be able to go from immunotherapy that is sort of one size fits all to something that is more personalized. And we've shown a scheme of how to go about and do this. And I didn't mention this, but also found an inhibitor that can be particularly potent in this setting to induce immunogenic malignant cell states in the background of immune evasion and genetic dysfunction. This goes hand in hand with high content mechanistic screens. So having the spatial output is going to be very important when we're trying to disentangle multicellular circuits as well as computational methods that will all go along with it. And I mentioned the RNA-based interventions because I think that this opens a completely untapped territory to overcome mechanisms of immune evasion that are based on deficits in RNA. And together, really thinking of how can we normalize immune responses as opposed to activating or suppressing them. And lastly, I started the talk saying that I'm going to talk about immune cell engineering, but I think that this is not quite the right term for what we're doing here. I think instead of rational design, what we've done was more to take the backseat and ask, can we leverage directed evolution to study biology, to discover new things, and to optimize cell functions in ways that maybe based on the knowledge that we have today, we won't be able to predict. And so with this approach, we think that we identified an underappreciated route of communication between cancer and immune cells, which is based on chemo-attracting metabolites. And we're very excited about the potential that it might have for new types of therapeutics. And with that, I want to thank all the members of my lab, our collaborators, funding resources, and thank you very much for your attention.
That was fantastic. Got time for one or two questions? Anyone?
Just so much like, I think the worry is with like some of these screens when you're looking at, say, regulators of cell cycle, is there any good way or ways that you guys, I mean, I know you were trying to give us a little overview with these metabolic sensors and metabolism in general that are going to come up as potential regulators to kind of split the hair between, is it mechanistic to the process versus just an indicator of kind of overall cell fitness, right? Do you know what I mean? Like,
so the metabolic component here is these receptors, we're not gating on anything that has anything to do with cell cycle, right? The NK cells are able to get recruited to the tumor when we put on metabolic sensing receptors on them, right? So whether or not this has other effects, this is something that we're looking into. So we've done some perturb-seq screens, for example, to see what happens to NK cells when they have these receptors. Turns out that it has some other effects as well, but they're actually better at, they're more cytotoxic, they have better effector functions. So it doesn't seem to interfere with that aspect, if that addresses your question, but yeah.
Have you looked at spatial metabolomics or lipidomics to see if these molecules are in the right vicinity and connecting with your GPCRs?
Yeah, we would love to do that. So we don't have the capacity to look at metabolomics quite yet, but I think that this is definitely needed. We wouldn't have, for a long time, we've been looking at the transcriptome thinking that it kind of tells us the whole story. But I think that based on what we're seeing with metabolite sensing receptors, we would definitely want to look at this spatially and getting a better understanding of at the micro scale as opposed to just, at the tumor scale in terms of recruitment.
Thank you, Livnat.
Thank you.